and welcome to the seventh talk of our Wonder Kids series. Thank you so much for joining us today. My name is Portia Queevy, and I'm the recruitment coordinator for the Early Development Research Group here at UBC. Speaking to us is Dr. Lauren Emberson, who, along with her wonderful team of trainees at the Baby Learning Lab, studies how young babies build an understanding of the world around them through their everyday experiences. Her research focuses on learning within the context of vision, language, and attention, and uses a combination of both behavioral and neuroimaging techniques. Dr. Emberson's Baby Learning Lab is one of 7 research centers that make up the Early Development Research Group. Because Dr. Emberson's lab is located on campus at the University of British Columbia, I would like to acknowledge that our campus is situated on the traditional, ancestral, and cedar territory of the Musqueam people. But of course, this is a virtual event, and so I would like to acknowledge as well the traditional owners and caretakers of the many lands that you may be joining from, both near and far. I will introduce Dr. Emberson in just a moment, and she will speak for about 30 minutes. Afterwards, she will answer your questions, taking us to about 2 o'clock. Please do feel free to pop in and out as you need. We understand that most of you have little ones at home. To gather your questions, we'll be using the Q&A window that you may access via the button along the bottom of your Zoom screen. Please submit your questions there and/or upvote questions from others you would like to have answered. If you're unable to stay for the entire talk or have technical issues, don't worry. We will be posting a recording of the talk on the EDRG website. I am delighted to introduce Dr. Emberson to you today, Assistant Professor in the Department of Psychology at UBC. Dr. Emberson gained her PhD in psychology in 2011 from Cornell University with a focus on perception, cognition, and development. Her research on the learning capabilities of the infant's brain, exploring how these abilities contribute to an infant's rapid development of perception, has been published in many top psychology journals, such as the Journal of Neuroscience and Proceedings of National Academy of Sciences, and as well as the Journal of, has been published in many top psychology journals, such as the Journal of Neuroscience and Proceedings of National Academy of Sciences. Dr. Emberson has also been the recipient of many prestigious awards, some of them being the Rising Star Award by the Association of Psychological Science and the Boyd McCandless Award. I'm sure her talk today will highlight why her work is celebrated within the field of developmental psychology. So not to keep you waiting any longer, please join me in welcoming Dr. Lauren Emberson.
Thank you, Portia, for that really, really generous introduction. And thank you all for being here. It is my pleasure to give you a little peek into our research in the field of baby learning. And so my talk today, if I can make the slides advance, yes, how do young babies learn? And learning, of course, is very linked to memory. So what I want us to do is to think back to our first memories, the very first ones you had as a child. And I want to share with you my first memory. My first memory, I think, was from around when I was 1.5, maybe 2, and I swallowed a nickel. I swallowed a nickel, and my dad held me up by my foot and hit me on the back, and the nickel popped out, thank goodness. But actually, the more salient part of my memory was the ice cream afterwards. My parents afterwards gave me strawberry ice cream. And while I love fresh strawberries, I actually don't like artificial strawberry flavor for whatever reason. I like other types of artificial fruit flavors, but I don't like artificial strawberry flavors. So I actually remember being a little bit peeved about the strawberry ice cream. So that's my very first memory, the nickel and the strawberry ice cream, and it was from around 1.5 or 2, I would estimate. But for your own first memory, how old were you? What was the age that you were? And often we find that first memories tend to be between 2 and 4. And the question that I have and that I want us to think about is, what about before that? We lived so much life. And for those of us who have little kids, they do so many things and they seem like they remember so many things when they're very little, even before they can speak. What's happening to all of those memories? So this is a phenomenon called infantile amnesia, or sometimes also called childhood amnesia. And here's just a little bit of background information on it. It was first reported by Carolyn Miles back in 1895, which is extremely old for the history of psychology. The psychology field really dates back to about this far, not much further than that. So this is a very old and classic phenomenon in psychology, infantile amnesia. And probably the more standard paper that people cite is Henri et Henri from 1890. And that paper reported that most people's first memories come between 2 to 4 years of age. And that, by and large, has been held up in the 100+ years of research on this topic since then. So 2 to 4 years is fairly standard, but there's also a huge range. So my memory tends to be, if this is a credible memory, is from before 2, and sometimes people's first memories are as late as 7. So there's a huge range there. And we know now that many, many factors influence early memories. Emotional context, that might be one of the reasons why my first memory is so young, is because I was choking, I was swallowing, I was put upside down, I was angry about the strawberry ice cream. My parents also, just as a bit of a personal side, they split up shortly after that. So I'm wondering if that might have also been why I had such an early memory. How personal the experience was, we tend to remember memories that are very much about us as opposed to something that happens to society. Like, an example that comes up often is Halley's Comet or other big societal events that happen that we all share. For some reason, we don't remember those as much. There's also a gender difference. Female versus male, they tend to see more early memories if you're female. And there's also big cultural differences. So there's just a ton of ways in which your early memories can be affected. And also some early memories may be reconstructed. So that's why I said about whether my first memory is real, is in part because they can often, you can think that they're your memory, but they're actually from family stories. So it's quite possible my early memory isn't my own memory at all. But that's a bit about infantile amnesia, but we're going to talk about learning. And the reason why this is connected is because one of the early theories about why babies didn't have, or we can't remember these early memories, is because babies didn't learn. They kind of didn't learn anything. And what they learned, they didn't retain. And in many decades of research that really started picking up around the 1950s and 60s, and that's the tradition that I'm continuing in my own research, we now know that babies are incredible learners. They learn in many, many different ways. And my part of the function of my research has been to look at the neuroscience of baby learning and how their learning is incredibly sophisticated at the level of their brain. So what were some of the ways we discovered that babies could learn? You think that that would be obvious, right? Obvious that babies learn. Now it feels obvious, but it wasn't at the time, in part because of the methods that we are using. And so one method that I'm going to show you now is called habituation. And this method has been used, by and large, this is the most important method in all of early developmental psychology, and it hinges on baby learning. So I'm going to give you an illustration that's inspired by summer in the Lower Mainland, summer in Vancouver. So let's take a look at this blueberry. We'll look at it for a little bit and then take it away. You can look at this blueberry again. Babies like looking at blueberries. Here's another blueberry. And now what do you prefer to look at? Got a blueberry, and blackberry season is right around the corner. I am so excited. And so what we see here, so habituation is the phenomenon where when you see the same thing over and over again, you start to become less interested in it. And that's true for adults, that's true for babies, that's true for non-human animals. And then after a period of time of seeing the same object, if you're given 2 objects, you often display what's known as a novelty response. So you prefer to look at the blackberry since you've seen the blueberry. And that's just a very simple type of learning. You can think of this in many different ways. And there's a number of different models that help us understand this type of baby learning. And one very simple one, I think this is quite interesting to think about in relation to the baby's behavior that you can observe in your own life and in babies that you know, is that if you only see blueberries a couple of times, babies prefer to actually keep looking at the blueberry. So this is called a familiarity effect. It's like they haven't quite figured out the blueberry thing. They're in it with the blueberry thing. They're wanting to learn more about the blueberries. You've showed me the blueberry twice. I still want to look at the blueberry. I don't care about this blackberry. But if you see the blueberry a lot and you're quite bored of the blueberry, now you want to look at the blackberry. So that's when you really see this very strong novelty preference. So this is one of the biggest, most classic findings in baby learning, that first of all, that if you present babies with one thing over and over again, you're going to see they become bored. And then there is this interesting complex pattern of what their preference is. Usually we look for these novelty preferences, but sometimes if babies haven't really finished their learning of the blueberry, they'll want to look at the blueberry again. And it turns out that habituation happens with many, many things. It's not just berries. I'm sure you're very surprised to hear that, but it's with faces and toys and actions. So all sorts of things that babies can be exposed to, all sorts of experiences in their lives, they show this simple type of learning. And this is true from birth, right? Very, very early on. They retain that memory, at least over the period of time of the experiment, and show these differences in learning afterwards. It also works with non-visual things. So it works with auditory studies as well, with the sound of your voice, the patterns of the language, the particular music that they're hearing, the sound of the dog barking. We can use habituation to study all of these things, and babies learn, in their auditory modalities with what they're hearing, their visual modalities, what they're seeing, with what they're smelling, with what they're touching. This seems like an incredible learning ability that babies can tap into very, very early on in life. And so like I mentioned, it's so important in part because we know that babies are now learning these sorts of things, at least over this short period of time, but also that babies, that we can use habituation to study so many things. So habituation is actually one of the main ways that we know a lot about baby cognition. What kind of individuals do they prefer? What kind of individuals do they remember? If they're experiencing a new toy, how many times do they have to see it before they recognize if the shape changes or the colors change? When they learn a new word, how long does it take them to learn it? We use habituation to study all of those things. I'll just illustrate in the word example. If I'll pick an example here, like say I teach a baby that this is a tuva, and I say tuva, tuva, tuva a whole lot, and then I say this is a dax. They'll go, whoa, that's different, and look longer because of the habituation to tuva and the object. So we use habituation to study baby cognition, and it's a type of learning in its own right. But it doesn't really feel satisfying when the answer comes to infant amnesia, because you might say, well, yeah, babies can do this type of learning, but that's not really the same as remembering, if something really like happened, like remembering the first time that you saw a horse if you were a 1-year-old. Like, why don't we remember that? That's not really habituation. Habituation could be just for a short period of time and you forget it. Well, that brings me to a really incredible researcher, Professor Carolyn Rovey Collier from Rutgers University, which is in New Jersey. And she was a real pioneer in arguing that babies actually do have incredible learning abilities and they retain those memories for a long, long period of time. And my work really builds a lot off of her work, and I'll explain sort of what the mobiles are behind her, as well as her classic experiments. So in these experiments, and I really think Professor Rovey Collier was such an incredible genius in how she showed this. What she did is she went to babies' homes, and she installed this new mobile that had these like really distinctive objects, and she had and she had the experimenters tie the mobile to the baby's feet. And so when the babies kicked, the mobile moved. And babies learned that actually quite quickly, that when they kicked, the mobile moved, because it's such an exciting thing for babies. A lot of folks who sort of do contingent baby motor things like this, like we put those rattles on their hands or their feet and they kick and they make the sound and they get really excited, or you can put a light toy on their hands when they're very little and they shake it. It's so exciting for them. And these babies, I mean, she did studies with so many different ages, but these ones would be around like 3 months age, quite young. And so they're kicking and the mobile's moving and it's so new and exciting and these bright contrasting colors. So cool. So babies learn that, and you can show that they learn it in a period of time similar to habituation. It's a more complex type of memory because they're learning the relationship between things. But what Carolyn Rovey Collier showed with these types of experiments was a number of really key things that really sort of raises the bar and increases what we think of in terms of the complexity of baby memory. First of all, they can retain this memory of this mobile and the kicking for months and months and months. So how she tests that is by going back to the baby's house, putting up the mobile again. This time there isn't a connection to the baby's foot, and she sees how much do the babies kick. And you can compare that to another situation where, say, there isn't a mobile or there's a different mobile, or it's a different context. And what you see is, when the babies are just see a mobile, they're not just going to kick randomly, but when they see that mobile, they've learned that connection, they kick a lot. Because they're remembering, hey, last time I kicked my foot and the mobile moved and it was so cool. That's the way that we can see that babies are remembering that relationship when they see the mobile again. She found that if babies can remember for many months, even when they're just 4 months old. And if you just simply show the babies a mobile again, you don't actually connect it with their foot, you just show them the mobile, they can remember. If you show them, say, every month, they can remember for almost years. She basically couldn't run studies long enough to show that babies didn't remember. So they've got this incredible memory. It's like this incredible paradox, right? How can they remember these sorts of things and yet we don't remember our first memories? She also showed that memories are context specific. So, you can see here the baby on the right, they've got this really big bumper around. If you have bumpers that are really distinctive, like bright, really dark colored stripes that are really visually salient to babies, that affects their memory in a way that if they had a different bumper, like imagine circles or something like that. If you change the bumper, it changes their memory. And that's actually similar to us. Adults are very, very sensitive to context in our memories, and particularly the memories that type of memories Carolyn Rovio-Collier was studying. I'm going to show you one last type of learning, and then I'm going to get into a little bit of my own research. The last type of learning that has been really a huge focus of the field in the last decade or so is called statistical learning. And this is kind of a blend between habituation and the sort of complex type of learning and memories that Carolyn Rovio-Collier was studying. And this one we think is very related to language learning. And so imagine, I have a bunch of words here. They're novel words to all of us. Imagine hearing them. It would sound a lot like a language that we don't know. But unbeknownst to us, these words, these little syllables are organized into words. So you can see them color coded here. So if, for example, you look at sort of the orangey one, you'll see it's meep, jik, dak, and it occurs again, meep, jik, dak. And it's kind of like the words in a language, right? We use syllables interchangeably across words, but they occur at a higher probability within a word. So the idea is that this is kind of like a baby learning a new language or learning their ambient language that will likely become their native language. How do they do it? How do they learn these patterns? This is a complex learning problem and the matter of a lot of debate. Theoretical debate in the field. How do they learn it? It's such a complex problem. Well, it turns out babies just have an incredible learning capacities and they can do it, and they do it by the statistics. So here, if you take one of the words out, so this is red league zet. If I expose babies to this language for a period of time, maybe 2 minutes, 3 minutes, just while they're playing with a caregiver or watching a video, we're not instructing them to learn. And we're not giving them any kind of feedback. They're just listening. And then afterwards, I can play them something that's consistent with the statistics of the language. So red league zet are syllables that tended to go together, kind of like a word. Babies will look for an amount of time, but then they'll be like, yeah, whatever, I heard that before. But if I present the baby with zet feng rao, they'll be like, what? Those don't go together. What are you talking about? Zet is at the end of a word. Feng is at the beginning of a word, and they look much longer. So again, we're using habituation to study learning here, but now we're looking at a much more complex type of learning. This is a learning of the patterns of syllables in a language. There's been so many studies, literally hundreds and thousands of studies on statistical learning in the last decade showing all the complex ways that babies can use this type of learning ability to tap into the basic structure of their language. And babies can do this starting at birth. We don't know too much before birth, but certainly by birth, they can, at full-term birth, they can do this type of learning, and they're tapping into the patterns of the language that the caregivers are speaking and they're hearing around them. One of the things that we've been doing in the Baby Learning Lab is looking at different types of statistical learning. So statistical learning, like habituation, occurs with many different types of experiences. Sounds, speech-like sounds, music, faces, actions, objects, all of those. How are they the same and how are they different? One theory says they're all the same. It's one type of learning ability and it doesn't really matter what you get because they can learn across all of these. Well, we've been showing they actually are quite different. So if you present babies with the same types of patterns in their speech or their faces, both of them babies really love to look at or listen to, what we find is that they learn both, but they are much better at 8 to 9 months of age at learning the patterns in the sounds with the speech than they are with the faces. We're doing a lot of follow-up work to see how does this relate to their emerging language abilities. At 9 months, babies are starting to, in some cases, produce their first words. They're comprehending more and more words all the time. We want to understand how the learning abilities that babies have are interrelated to all of their other abilities, as well as understanding in a really deep way the mechanisms that are underlying these. So we can understand, based on the experiences that a baby has, what they're likely learning and how that's contributing to their development. The other sort of contribution of my work is I do a lot of baby neuroimaging. And this is what it looks like. We use very infant-friendly neuroimaging techniques. We have these very light caps, and these caps have lights embedded in them. And they're similar to pulse oximeters that babies would have, for example, in the NICU to monitor the oxygenation in their bodies. And this is a technique called functional near-infrared spectroscopy. And one of the reasons why this is such an incredible technique is that it gives us a little peek into how the baby brain is responding while they're learning, while they're experiencing something new, which is something we've not really been able to do before without this technology. As you can see that this baby here is able to read a book, interact with their caregiver, see all these new patterns and experiences. So using FNIRS, what have we found? Well, in one study, we taught babies that sounds went with new objects. You can think of this kind of like learning a new toy, right? You get this new toy, it looks a cool way, and it makes a new sound. Like sometimes they rattle, sometimes they squeak, and we can teach babies this really, really quickly. So that's what's on the right, the audiovisual trial. That gives you all the details in terms of the timing and how it all worked out if you're interested. And while babies learned this new pattern, about this new object that popped up on the screen and made a cool sound, we also did recordings at 3 different parts of their brain: the frontal lobe, the temporal lobe, and the occipital lobe. And you can see a baby. This is little baby Wilhelmina who's in the study here. She's such a cute cutie peanut. So we have the frontal lobe is a really important part of the brain. It's a part of the brain that actually shows changes, developmental changes through adolescence into your 20s. This is often the part of the brain we think of as being very important for high level cognition, inhibition of activity. People talk about the frontal lobe being like the most important, most sophisticated area. So we're recording this in 6-month-old babies. The temporal lobe is a part of the brain very important for language and listening, the auditory system. And the occipital part of the brain in the back is very important for vision, among other things. So we're parting the auditory part of the brain because there's sounds involved, the visual part of the brain because there's visual stuff involved, and the frontal lobe in part to see what would happen. And what we found was really quite astonishing, that while babies learned this new sound and object, they participated in just about 3 to 5 minutes of watching this. We saw substantial changes in every single one of these regions. Every single one of these regions was involved in learning. It was changing its profile of activity while babies were learning these new sounds and events. We also have found some evidence that actually the frontal lobe is really key in learning here. And some other labs are finding this as well, that even though we think of this part of the brain as being something that we use more and more as we get older, like adolescence, and important for all of these higher level cognition things, babies are using this part of the brain to learn. When they have a new experience, their frontal lobe is involved in telling all these other parts of the brain, like, hey, there are these cool patterns happening out in the world. You guys might want to check this out. It's one way of thinking about what it's doing. It also is really important in saying when something changes. So if it picks up on a pattern initially, and then we change it up, the frontal lobe is like, whoa, guys, you have no idea. All of a sudden, the pattern is different. The frontal lobe is really involved in learning with very young, even young babies, 6 months old. Really incredible finding for us. So many parts of the baby's brain are involved in learning, not just like parts that are really involved in the sensory part of it, not just the visual part, not just the auditory part, but the frontal part too. And they're all working together. And it's happening really quickly on the matter of minutes. The next thing, so I'm going to dive in and get pretty sciencey here for a minute. I'm going to show you a graph. So bear with me here. I'm going to walk you through it. So the other thing that we wanted to see was how much does the brain change? How much does learning change the brain, the profile of particular responses? So this is something that we think a lot about in cognitive neuroscience. What is the profile of response of a part of the brain, the sort of specialization of the part of the brain? So when we read things like this part of the brain does this, or this part of the brain does that, that's all about looking at the sort of profile response. This part of the brain responds to faces, not to houses. I don't know if anyone followed the Jennifer Aniston cell that came out many, many years ago. It's this, part of the brain responds to a picture of Jennifer Aniston, for those of us who are of my generation and watched Friends the first time, would know all about that. So anyway, this is showing the profile of responses for the two parts of the brain on the left. The temporal lobe in blue, so remember I said that's a very auditory part of the brain, and the visual part, or the occipital lobe, excuse me, in red. And what this profile is basically just showing is what we would expect, which is that the auditory part responds to the auditory stuff and the visual part responds to the visual stuff. This is like a duh, kind of like just go home, not interesting kind of plot. And this is what we'd expect in adults. This is what we'd expect in babies. We're just showing that that's the case, with some new sounds and visual events that occur on their own, like just the sound on its own temporal lobe, just the visual on its own occipital lobe. And when we put them together, not too surprisingly, they get the sounds and the visual events together. Remember, they're learning that like this new toy makes a sound. Both areas are active. So it's like, oh yeah, I've got sound and I've got vision together. I'm learning them together. And you can see that there's actually a lot of activity when they're learning. So not only do they both show together when they're learning, as we'd expect, but the activity goes up. These parts are really, really active when they're able to learn that things go together. So the baby brain is engaged. Then what we did was we changed it up. Remember I mentioned that the frontal lobe and the brain really cares when they learn a pattern and then we switch it up. It's something new. It's kind of like after seeing the blueberry for a long time, I show you a blackberry. Your brain is like, whoa, something different, something cool. So what happens? Babies have learned the sound and the visual events go together. They've learned about this new toy, and now I'm just going to present the sound. What happens to the visual system? And this is going to tell us a little bit about how much the brain has changed with learning. And what we see is that when they just play the sound on its own, that the visual system really changes a lot once babies have learned. And I don't know, Portia, can you see my mouse here? I should have asked before.
Yes, see, I can.
Yeah.
Yes, I can. Yeah.
Awesome. Thanks, Portia. I hope you can all see my mouse. But what's crucial to look at is here. Sorry for diving so sciencey here on a Saturday. We shall be kicking back, having a beverage of our choice. But this is something that gets me up in the morning and makes me so excited about all the research we do at the EDRG, is that with just a couple of minutes of learning, we actually change the whole profile of this part of the brain for a period of time for this particular study, for this particular object. We're not totally changing babies' brains here or anything, but what it showed that when babies learn something new, if you present just a sound on its own, normally this is what it looks like. And the occipital lobe doesn't care. It's not a visual thing. But when the baby has learned about this new toy that has sounds and visual stuff that all go together, it's so cool. All of a sudden, you get this big response. And so what it's showing is that new experiences change the baby's brain really quickly and really dynamically. The baby's brain is so incredibly sophisticated in how it responds to these new experiences and just everyday experiences, like learning about a new toy. This isn't anything like taking your baby to the top of a mountain. This is just, here's a cool new toy, the brain is all involved and changes how it responds. And we think that this is a little peek into the bigger picture of development, of how babies are learning all of their experiences in their lives to grow and change and develop. So let's just wrap up here. I'm going to start to think about the big picture. How does this relate to these first memories that I talked about? Well, I think it relates in two ways. First, this like question about infantile amnesia really motivated and has motivated scientists for decades and decades and decades to ask about baby learning. What is baby learning doing? What's it like? And what's happening with these memories? Now, we know much, much more about baby memory and baby learning, and we still don't really have a solution as to what's happening to infantile amnesia. We have a lot of different answers, but we don't have one answer. We're still working on it. But it's told us so much more and so many things that are so important and really enriched the lives, not just of scientists, but of parents in trying to understand what's happening with their babies, what's happening with their minds, how are they developing, and helping them to engage and to be part of the developmental process in that intellectual way as well. And also, what is this sort of doing for the world? This is one of the things I want to motivate, especially these day and ages. We're all thinking about how can we not just do something in sort of the ivory tower, but what does this mean for everyone? And for my lab, we do lots of work looking at even how very, very young babies are learning. So the first picture here on the left is a neonatal baby who's having the neuroimaging happen with the cute little cap. And so we're looking at how do the youngest babies learn. Babies when they're first born, when they're full term, but also preterm babies. And that's the second picture here. We started doing some recordings with babies in the NICU, and we're also looking at how preterm birth affects learning and neural development. When you're born very and extremely preterm, you can be at risk for problems later on. And what we want to understand is why and how we can help. So we have a number of studies to that end, and we're collaborating with BC Women's and Children's Hospital with clinicians there to apply what we know about the theories of development, theories of neural development and cognitive development and learning in babies to help unravel that puzzle and to provide better care and support for that important population. The lab also does work with the Bill and Melinda Gates Foundation, looking at development across the world, and particularly looking at babies who are being raised in highly impoverished contexts, where we know that they're more likely to miss developmental milestones. What's happening in their brains, and how can we understand why, and how can we also come up with targets that are really sensitive for interventions? If we intervene on prenatal health, we intervene on the family context, how can we show that that's having an impact on the baby's development, and particularly their neural development? So there's lots of ways that outside the laboratory, we think this work is helping make the world a better place and help babies develop to their full potential everywhere in the world, including here in BC and also abroad. The other thing that's great about this research is it's so fun. I do want to thank all the families who volunteered for the research that I presented, and also show a little bit what it's like to participate in an EDRG study. Of course, this is a picture pre-pandemic. We've been shut by and large through most of the pandemic, but this fall we'll be opening again. It won't look exactly like this. It'll look a much safer version of this, but it will, I promise you, it will still be very fun. And we really hope that some of you come in and join us. We are all very excited to see families again. And in the meantime, we have online studies, which are also quite fun. My kiddos participate in them, and I can say my older one is often very jealous, but the little one has one just on her own. But the researchers are also amazing at involving the older one, so she doesn't feel left out. So it's another way to be part of the EDRG, even as we're transitioning to opening up and the reopening of everything in BC starting this fall. So here's exactly how to sign up to participate. We're going to show this again at the end. So this takes you to the EDRG website, so you can put in your information so we can contact you and your family when you're eligible for studies. And I also want to point you to all the previous incredible Wonder Kids talks. If this happens to be your first, I am honored, but I certainly encourage you to look at all the others. Dr. Janet Worker, Dr. Kylie Hamlin, Jeff Hall, Darko Odik, Andy Barron, and Sue Birch, all on different topics, all the research that we do in the EDRG. They're all really amazing, and we'll tell you a lot more about your baby's and child's development. So thank you so much from all of us at the UBC Early Developmental Research Group, the EDRG. And it's time for Q&A. I would love to answer all of your questions. So please put your questions in the Q&A box, and you can vote on questions that you would like to see answered. Thank you.
Yeah, thank you so much, Dr. Emberson. So yeah, just like Dr. Lauren Emberson said, she's more than happy to answer some of your questions now. So if you haven't already, please use the Q&A button at the bottom of your Zoom screen, and you may view questions others have already submitted and vote for the ones that you would like to have answered. So feel free to ask any questions that you may have about the research that we do at the EDRG, or anything that Dr. Lauren spoke about during the presentation. But if you also have no questions, that's okay.
Portia, do you have a question?
I think I do. I was really fascinated about the study where the babies have the the mobile hanging at the top, and then they have it attached to their legs and they kick it, and they get this, they learn that they when they move their legs, the mobile moves. And you, I think you mentioned that it's specific to the specific mobile. So do they learn that this particular like object, it's like, this only works if they kick their legs for the mobile that has this particular object, or is it for any mobile that like hangs over them? That's really fascinating to know that.
That's a great question. Yeah, they learned that it's the specific ones. It depends in that, what Carolyn, like all the research that Carolyn Roby-Collier did, had these very distinctive mobiles that wouldn't have been, in that day and age, wouldn't have been what the style of mobiles would have been. The style of mobiles now are actually are a lot closer to those, right? With often like high contrast objects. That's kind of the style of baby toys now, informed by developmental research, actually. Developmental research is what has shown that babies see those things, bright colors, stripes, black and white very well, that you see in baby toys now. But that wouldn't have been the style of mobiles then. So baby saw this extremely distinctive mobile with these particular shapes. And yeah, she found that if you have another extremely distinctive mobile, but with different shapes, they don't kick. So they know that it's fairly specific. Yeah. So that's one of the things she was trying to show, that it was this, she wants to argue that the hippocampus was very active in babies. It's a particular part of your brain you may have heard, folks may have heard of, for a particular type of memory. And part of the importance there is a specific memory. And she had studies that would argue that.
That's really fascinating, because then they would have to remember that this specific object with this like color difference and all of this is the one that will move when your legs kick. That's impressive.
Exactly.
Baby's so young.
Isn't that amazing? I mean, I feel like I wouldn't remember that.
I would not. I would not.
I will say that some of the conclusions of Carolyn Roby-Collier's work in terms of the hippocampus being active are really still very debated today. Her findings are not that babies can do all these types of memories. We've sort of moved on as a field to start to ask, well, which parts of the brain are really active? Some people think that it is the hippocampus, the way Carolyn Roby-Collier argued. And other people would say, no, it's probably using different parts of the brain. But the specificity is something, and all of the fact that babies can retain it for so long is all still very I don't want to say sound research, because it was sound even at the day, but like not currently part of the debate. But we continue to build from those findings to understand more and more about how the baby brain learns and forms memories. But the specific ones.
That's really interesting. Yeah, very, very interesting. Very interesting. And I think maybe another question that I have is based off of your research. Are you looking at how babies, like what kind of memories stay the longest in babies? Because I do know, like, for example, I've read of how emotional memories for adults and kids have some of the memories that are ingrained so well into our minds. And is this something that begins from the baby, from a young baby's age, or is it something that is learned over time? If you can see.
Yeah. We haven't, most of my research has been on these short-term effects of learning, not so much the long-term effects of learning, but some of our projects that we're hoping to start up in the fall are going to start looking more at the long-term effects and specifically trying to bridge the two. Generally, the field of development has looked, and just broadly speaking, has really separated them both, right? Separated these two out, I should say, where you have people who are studying short-term memory, like learning effects, like I do, and really long-term memory stuff, like either asking you to remember your first memories, or showing you something and bringing you back days, weeks, months later to ask, do you continue to remember? But very little work is bridging the two, and particularly in infancy work. And so that's one of the things that we're hoping to do, is to teach babies something new and see how long they retain it, and see what helps them retain it longer. This in some ways is very similar to Carolyn Roby-Collier's work, but we're going to do it with different types of learning. Her type of learning with the sort of kicking and the contingency is a special type that we think is very likely to stay for a long time. But we want to be looking at more of this sort of pattern learning, more like statistical learning, and see how long we can get babies to remember that. And this is part of that, the bigger story there is trying to understand how something like statistical learning helps build language abilities. It helps build visual abilities, right? When we when we learn a language, we don't just forget it right away, right? Like,
Mhm.
I mean, if for me, as someone who's learning a second language, I can tell you as an adult, it does feel like sometimes you forget it right away. But when we build our language knowledge, it's over time and over time, and same with babies. How do these short little changes that we see in the lab build and build and build into these concrete abilities that they're ultimately using in their lives? That's
Yeah.
something that we really want to understand, because we think that that process might be affected if babies are being born preterm, or if they're
Mhm.
at risk for a variety of ways, and we might be able to pick up on those changes of that changing from that short-term learning to the long-term memory might be different in those babies, and then we might be able to intervene, we might be able to help, we might be able to show that they should get some more support, early intervention, or come up with novel ways of helping them.
Yeah. Thank you so much for that. We do have one question in the chat, and it's what, if any, what are, if there are any measurement differences between EEGs and FNIRS, and it's now EEGs for adults, while FNIRS is for infants. So is it EEGs are for adults only, and functional NIRS are actually for infants?
That's an amazing question. Thank you. Thank you so much for that question, audience member. Yeah, so actually the people do a fair amount of EEG with babies as well. EEG with babies is pretty challenging, in part because EEG is very sensitive to motion, and it's kind of hard to tell a 6-month-old to stay still. You can say it a lot, they kind of don't listen. So, and when you blink and all sorts of other things. So I have some very close colleagues who are EEG and collaborators who are EEG researchers with babies, and I feel like it's heroic work. But there is EEG with babies. It's actually one of the most established neuroimaging techniques. EEG is extremely common in adults, so is fMRI. Where FNIRS fits in as a relatively new modality is there's a lot of FNIRS in babies, in part because it's very hard to do fMRI in babies for a variety of reasons. FNIRS is very similar to fMRI. The sort of similarity in the name is not just a coincidence. It's actually, there's a lot of parallelism in the techniques. And I would say that basically it's fMRI with babies. We do it in an optical way using lights as opposed to with a big scanner, an MR scanner, which is one of the reasons why it's much better for babies, in my opinion. Babies don't need to go into a scanner. They can sit with their parents. It's very light. There's fewer risks, etc. You can do FNIRS with adults. There is a big field of people who are doing it. I would say in my hands, I find it quite challenging. It's a lot of specificity you need with the technique, and the technology is developing tremendously. But one of the things that's really great about FNIRS with babies that helps with adults is that the number of people that you can do recordings with is much bigger. So I'll give you an example. Say you wanted to study anxiety disorder and you didn't want to use EEG. How many people with an anxiety disorder are going to go in an MR scanner? If anyone here has had an MR, it's like this long tube. You tend to feel very claustrophobic, and it's an incredibly safe and incredibly important technique. I don't mean to say anything bad about it, but it does limit the populations who are going to go in it. And you can kind of go on from there. Anxiety disorders, autism, someone with a cochlear implant, because there's electronics in the cochlear implant, they can't go in the MR scanner. So NIRS is a really important technique for reaching other populations that they can't traditionally go in the MR scanner. And I think that's one of the things that's so important as a field, is that we become more and more inclusive and understand more and more about the diversity of the human brain and mind, and how we can use neuroscientific tools to help. My one last pitch too for adult FNIRS is that there are some people who are using adult FNIRS to do really incredible work about understanding the real kind of our real life contexts. When you're in a scanner, you can't do something like look at another person, play an instrument, but you can actually use FNIRS to study how does the musician brain operate when they're playing with someone else? How does the brain respond when you physically look at another person or talk with them? And so it's a challenging technique in adults for a variety of reasons. I can kind of go into that if the person is interested, like skull thickness and other stuff can make a difference there, just because adults are bigger. But I really think that work is going to push in incredible ways, push the boundaries of what we know about the mind and the brain. Thank you for that question. That was a great one. It clearly got me on my soapbox.
Thank you so much, Dr. Emberson. We're nearing 2 p.m. So thank you so much for being with us today and for taking us through your research and teaching us so much about how babies learn. And it's really fascinating. It's a really sunny day outside, so I hope everyone who joined us today can go outside and do a little something, enjoy the sun and maybe go to the beach, in a safe way, of course. Thank you all so much, and we hope to see you again at our next talk later in the fall. Bye.